Assalamu alaikum, dear students. Hope you all are fine. I am Dr. Maham Steek from Botany Department, University of Education, working as a co-teacher in subject of plant systematics, anatomy and development. Course code is BOTN 1112. Today we will discuss structure and development of root. As we all know that root is the crucial part of the plant. In this lecture, we will discuss anatomy of root, in which root cap, epidermis, cortical tissues and vascular tissues will be discussed. In addition to these tissues, lateral roots, structure of dicot root, monocot root and their differences will also be discussed. What is root? Root is the underground part of the plant. It anchors the plant in the soil. It is also used to absorb water and minerals from the soil. The root structure is almost uniform throughout its length. It is without nodes and internodes. There are two types of roots: taproot or primary root. Second one is adventitious root. Root has three distinct tissue systems. These are epidermal, cortical and vascular tissue systems. This diagram shows the cross section of a root. In this diagram, you can see the outermost layer, epidermis, from which root hairs arises. Next is the ground tissues. Ground tissues include the cortex and the endodermis. Next is the vascular tissues. Vascular tissues present in the center. These are located primary xylem in the red color and primary phloem shows by yellow color. Pericycle is a thin layer of plant tissue between the endodermis and the phloem. On the other hand, we can see diagram of root structure with their cell structure. In this diagram, you can see the epidermis and root hairs arises from the epidermal cells. Then cortex. This is the cortex. Blue line show the endodermis layer. Then xylem and phloem shows vascular tissues, and pericycle, as we defined earlier, it is the thin layer present between the endodermis and the phloem. Next, you can see a word stele. Stele is the central part of the root, including vascular tissues, and in some cases ground tissues and pericycle, which, if present, define the outer boundary of the stele. Next is root cap. Root cap is present at the tip of the root. It protects the underlying apical meristem. Root caps also help in penetration of root in soil. Root cap also control the geotropic response of root. The cells of the root cap are living. They contain abundant starch grains. The walls of the cells are mucilaginous. Mucilaginous walls help in gradual sloughing of cells from the outer layer. Apical meristem adds new cells and increases the thickness of root cap. Central cells of the root cap in many parts form a constant structure called chlomela. Environmental conditions stimulate the development of root cap. Root caps are not developed where plants are grown in solutions. Root cap consists of parenchymatous cells in various stages of differentiation. It is protective in function. Recent experiments have indicated that the root cap has another function that is physiologically of great importance. Root cap is apparently the site of the perception of gravity. Thus, it appears to be capable of controlling the production in the meristem of the growth, regulating substances involved in geotropism or their movement. However, the root cap itself is not the site of synthesis of growth substances. The experiment shows that the root cap can control the movement, if not also the synthesis of endogenous auxins in the root apex. This is the longitudinal section of root tip. In this diagram, our only concern is about root cap. As we discussed before, root cap is protects the underlying apical meristem. Root caps help in the penetration of roots in the soil. And in this diagram, you can see the light mustard shade indicated the epidermis, and the center of the root cap consists of some constant structures called chlomela. This is the quiescent center of the root cap. Next is the epidermis. Epidermis is also known as epiblema or piliferous layer. In most of the root hairs develop from some of the epidermal cells at a little distance from the apical meristem. In most cells give rise to root hairs are known as trichoblast. Trichoblast is usually distally situated cells of this gene. Root hairs are outgrowth of single cells and function both in absorption and in anchorage. Root hairs are usually eventually sloughed off, but occasionally they are persistent. Electron microscopy reveals that wall of root hairs is a continuation of only the inner component of the walls of the epidermal cells which give rise to it. In some species, root hairs are formed from special cells which are distinct in size and metabolism from the neighboring epidermal cells. These cells are known as trichoblast. In this diagram, see that this is the structure of epidermis of root. This is the epidermal cell from which root hair is growth. The outer wall is cell wall, then cell membrane, The inner portion is cells, cytoplasm, some vacuoles and nucleus is present. This part show the formation of root hairs. Full grown root hair is seen in this diagram. Next, cortex. Cortex. In most roots of cortex is parenchymatous. In some roots, the cells of the cortex are very regularly arranged, both radially and in concentric circles. The cortical cells contain starch and sometimes crystals. Sclerenchyma is more common in the roots of monocotyledons than those of dicotyledons. Collenchyma is occasionally present in cortex. Next is endodermis. Endodermis comprises of single layer of cells different physiologically and in structure and function from those on either side of it. In the young endodermal cells, a band of suberin Casparian strips run radially around the cell and is thus seen in the radial walls in the transverse sections of roots. This suberin deposit, to which the protoplast of the cell is attached, is continuous across the middle lamella of the walls, but is absent in the tangential walls. A study of endodermis with the electron microscope by Bonnet 1968 reveals a thickening of the wall in the region of Casparian strips. The plasmalemma is thicker here and adheres strongly to the cell wall. The thin-walled passage cells often remain in the endodermis in position opposite to protoxylem. In this diagram, epidermis is present at the outermost layer and root hair arises from this epidermis layer. Then endodermis is present, and between the epidermis and endodermis, cortex is present. In endodermis, Casparian strips are present. In other diagrams, Casparian strips are present in the endodermis. Endodermis is in blue color and black color shows the Casparian strips. Next is pericycle, vascular tissues in which xylem, phloem included. Pericycle is usually a single layer of parenchymatous cells lying just within the endodermis and peripheral to the vascular tissues. Pericycle has a capacity for meristematic growth and give rise to lateral roots, parts of vascular cambium, and usually the meristem which produces cork and phallogen. Pericycle is sometimes called procambium. Vascular tissues. Vascular tissues included xylem and phloem. Xylem forms a solid central core in the gymnosperm and dicotyledonous roots. The outer margin of the central core has ridges. These ridges extend towards the pericycle. The number of xylem ridges varies in different plants. Protoxylem elements at the tips of ridges are narrow. They have reticulate annular or spiral thickening on their walls. Xylem elements are wider in the center. It contains metaxylem. This type of arrangement in which protoxylem lies towards the outside and metaxylem lies towards the inside is called egg arch. Next is phloem. Phloem strands alternate with ridges of the xylem and have the same number. In each phloem, the protophlloem elements are present towards the outside and metaphlloem are present towards the inner side. Phloem consists of sieve tube elements, parenchyma cells, and few fibers. Cambium. A parenchymatous sheet of tissues separate the phloem strands from the xylem. It becomes meristematic cambium at the start of the secondary groove. In the center of the vascular tissues, pith is present. Few parenchyma cells are present in the center of the xylem core is called pith. In this diagram, you can see the whole structure of the root. First of all, first layer is epidermis, which is called pelliferous layer, from which root hairs arises. Then cortex is present. Cortex has parenchymatous cells. Sometimes collenchymatous cells are also present, and sclerenchymatous cells may or may not be present. Then there is a layer of passage cells, Casparian tip present in endodermis. Endodermis is the innermost layer of the cortex. Then pericycle, which surrounds the vascular tissues. Vascular tissues included phloem and xylem. Xylem is of two types, protoxylem and metaxylem. And in the center, pith is present. This is the ground plane of the diagram: root hairs, pelliferous layer, cortex, endodermis, pith, and in pink and blue, stones indicated the xylem and phloem, which is alternate to each other. Lateral roots. Lateral roots arise endogenously from other roots and increases the absorptive surface of the plant, sometimes referred to as secondary roots. Lateral roots ultimately mimic to a large extent the structure of the roots from which they originate. In this diagram, you can see the primary root, and from primary roots, lateral roots are arises, and on lateral roots, root hairs are arises. This is the root tip and root cap. In higher plants, angiosperm and gymnosperm, the development of the lateral root is commonly initiated by the pericycle, an intrastellar ground tissue of the parent root, and the lateral root ultimately makes its way by piercing the cortex and the epidermis. In lower vascular plants, the branch roots originate in the endodermis. The method of formation of lateral roots in higher plants is: a few mature cells of pericycle, usually opposite to the protoxylem group, become meristematic and go on dividing periclinally and anticlinally. You can see in this diagram. Thus, a number of cells are produced which form something like a protrusion. This is really the primordium of the lateral root. It soon takes the shape of growing point with its initial cells, the cap and the other histogens. With gradual development of the primordium and the other tissues surrounding it gets stretched and ultimately ruptured. This is how it eventually comes out, piercing all the tissues. As regards the mechanism of growth of the lateral root, some workers have suggested that it partially digests the cortical tissue during its advance, whereas others are of the opinion that it is entirely a matter of mechanical penetration. It has been reported in some cases that the endodermis also undergo anticlinal division and form a layer of surrounding the lateral root primordium, and by further periclinal division it may be even more than one layer in thickness. But at any rate, those cells die and are shed when the lateral root comes out. Structure of monocot roots. The outermost layer of the young roots, which has thin-walled cells, are called epiblema or epidermis. Some of the cells of the epiblema give rise to the root hairs. Both epiblema and the root hairs are devoid of cuticle. In older parts, epiblema is shed. Next portion is consists of cortex. Cortex encloses the intercellular spaces for the exchange of gases. The cells store food. Cortex of monocot roots have important three functions. First, conduction of water from the root hairs to the inner tissues. Storage of food. Third, the outermost layer of the cortex produce protective exodermis in the older roots. Next layer is endodermis. The brown layer is called endodermis. Endodermis is the boundary of the cortex and is single layered. It is made up of barrel-shaped cells which do not enclose intercellular spaces. Next to endodermis, pericycle is present. Pericycle or outer boundary of the vascular strands lies just below the endodermis. Pericycle may be uniserrate, means single-layered, or multilayered. In monocots, pericycle does not form cambium. It only produces lateral roots. After pericycle, vascular strands are present. It is in the form of several, 8 or more, as seen in this diagram. 1, 2, 3, 4, 5, 6, 7, 8. Alternate and radial xylem and phloem bundles. These are phloem bundles and these are xylem bundles. We can see these are alternate to each other. In many cases, vascular bundles are embedded in the slender of sclerenchymal cells. Vascular bundles are arranged in the form of rings around the central pith. This is central pith, and vascular bundles are arranged in the form of a ring. Xylem bundles are X-arch, as we discussed before. That is, the protoxylem lies towards the outside while the metaxylem faces inward. Phloem bundles alternate with the xylem bundles. There are two separated from each other by means of narrow strips of conjunctive tissues. These cells of this tissue store food if these cells are parenchymatic. In the center of the monocot root, pith is present in this region. It consists of parenchymatous cells which may be rounded or angular in shape. Intracellular species are present amongst the pith cells. Pith cells also store food. Next is structure of dicot root. Dicot root has also epidermis layer, single layer composed of thin walled cells and which is not cutinized, and some of the epidermal cells arises root hairs as in monocot root. Next is cortex. It is quite large and extensive in roots. Cortex is made of thin-walled, living parenchymatous cells with leucoplast, which convert sugar into starch grains. The last layer of the cortex is called endodermis. It is of universal occurrence in the roots. Endodermis is composed of one layer and have barrel-shaped cells as in the monocot roots and are closely arranged without having intercellular spaces. Next, central cylinder is present, endodermis. There is a single layer of cells called pericycle, which are made up of thin-walled parenchymatous cells. Pericycle arises the lateral roots. Vascular bundles are typically in the form of radial roots. Xylem and phloem form separate patches as shown in this diagram. Phloem in blue color and xylem in red color. Xylem has protoxylem towards circumference, metaxylem towards the center, and again this arrangement is called X-arch arrangement. Phloem with safe tube form patches arranged alternate with the xylem as in the monocot root. A small patch of sclerenchymal cells is also present outside every group of phloem. These blue colors. Conjunctive cells are thin-walled parenchymatous cells lying in between xylem and phloem. At the center, there is a small parenchymatous pith. It may be even absent in some dicotyledonous roots. Next is difference between monocot and dicot roots. Basis of comparison is on the base of their tissues. Monocot root and dicot root. First, cortex. Cortex may be heterogeneous with outer dead exodermis. Dicot root have cortex homogeneous, that is without any differentiation. Next is pericycle. In monocot root, pericycle only produce lateral roots, while in dicot roots, pericycle give rise lateral roots, cork cambium and some parts of the vascular cambium too. Next is pith. In monocot roots, pith is large, well developed, contain abundant amount of starch grains. While in dicot roots, pith is small or completely obliterated. Next is xylem and phloem. Xylem and phloem are numerous in number in monocot roots, while in dicot roots, xylem and phloem are limited in numbers. Next is passage cells. Usually, passage cells are absent in the endodermis of monocot roots, while passage cells are present in the endodermis of dicot roots. Next is secondary growth. Secondary growth is absent in monocot roots. Why? Due to the lack of vascular and cork cambium. As compared to monocot roots, secondary growth is present in the dicot roots due to the presence of vascular and cork cambium. Phloem fibers or phloem parenchyma completely absent in monocot roots. Phloem fibers are completely reduced in dicot roots too. Casparian strips very much present in the endodermis of the monocot roots. While in endodermal cells near protoxylem, no Casparian strips present. Next is conjunctive tissues. Conjunctive tissues are limited or even absent in monocot roots, while these are well developed in the dicot roots. Metaxylem elements are oval or circular, while in dicot roots, metaxylem elements are angular, arranged in the linear position. Thank you so much.